Hello everyone, my name is Broder Thurston and I'm currently a third-year health sciences student here at Carroll College. Now before I begin, I would first like to thank everybody for coming and observing my presentation. Now today I will be talking to you about my research regarding the mechanistic effects of vitamin D3 on human breast cancer cell lines. So what do I mean when I say mechanistic effects? With this, I'm referring to the induction of cell apoptosis, which is programmed cell death among cells, cell proliferation, which is essentially cell division of one parent cell dividing into two daughter cells, and cell growth inhibition, which is the reduction or complete stop of cell growth. Now for some background information regarding breast cancer entirely. Out of all the women in the United States, 1 in 8 are expected to be diagnosed with invasive breast cancer. In 2021 alone, about 330,840 women are expected to be diagnosed with invasive breast cancer. Now when I say invasive, I'm referring to the spread of cancer cells into the tissue of the breasts. Now we all know that the most common form of treatment for breast cancer is chemotherapy and radiation. These are two treatment options which have been around for a really long time. But now there's current research showing that vitamin D3 and its analogs prove to be a viable treatment option for breast cancer. The active form of vitamin D3 is calcitriol, and when it is used in vitro, cancerous cells have been shown to have a greater rate of cell apoptosis. Now going off of that, calcitriol has shown effects of stimulating pro-apoptotic cells as well as repressing anti-apoptotic proteins in various cancerous cells. Now the overall purpose of this study was to determine if vitamin D3 shows promising mechanistic effects for the rate of cell apoptosis within these cancerous cells, as well as its ability to act as a tumor suppressor in comparison to traditional chemotherapy and radiation treatments. Now, based on the findings of this research, the doors can be opened to a wider range of treatment options for women with breast cancer, rather than just the traditional treatments of chemotherapy and radiation. So to get started on the research aspect, I performed a systematic review which consisted of 7 primary sources of literature. In order to obtain the literature, I used database searches from PubMed and CINAHL in January and February of this year. Before inclusion criteria was applied, there were 52 articles that I found, and I was able to narrow those down to 7 studies which were used in this systematic review. And when we look at the results, it was concluded that vitamin D3 would in fact be used as a viable treatment option for treating breast cancer cells. A majority of the literature focused heavily on the MCF7 cell line, which is a very aggressive breast cancer cell line. And based on the lab studies, vitamin D3 and its analogs have shown promising anti-proliferative effects and apoptotic results among breast cancer cells. If we look at figure 2 here in the middle column, we can see 2 breast cancer cells. On the left, we have SUM 149, which is another breast cancer cell line that did not undergo any type of intervention. On the right, we can see the same type of cell treated with calcitriol. It is clear to see the size reduction. And all in all, vitamin D3 has demonstrated the ability to induce cell apoptosis among breast cancer cells and inhibit cell growth. In one study, mice were used as the test subjects, and within the study, the mice were treated with calcitriol, and that showed cancerous cell reduction by 60%, which corresponds to the information shown in figure 2. So what else does this look like? If we look to figure 1 in the lower left corner, we can see the densely packed MCF7 cells. This image was taken with no vitamin D3 intervention. You can see that the cells are intact and the chromatin in the nucleus are unfragmented. If you look to the right at figure 3, you can see that the same cells, the MCF7 cells, are showing signs of damage. Within the cell nucleus, the chromatin is fragmented, and this suggests that the vitamin D binding protein did in fact induce cell apoptosis. Now, as I said before, most of the studies that I used did find a favorable correlation between the intervention of vitamin D3 and its analogs in the effects of breast cancer cells. Now, I know that we talked about the MCF7 cell lines and their results already. However, in one study, there was one cell line, which was the MDA-MB231 cells, that did not find any changes when treated with calcitriol alone. However, when it was treated in conjugation with another treatment option, it did show significant cell growth inhibition and apoptosis. Along with this cell line, we saw results in the SUM 149 cells, the MCF7, LNCAP, which all showed significant cell growth inhibition, apoptosis, and cell proliferation. Now, Among all of these results, it was proven that the vitamin D3 interventions seemed to be most effective when treatment of cancerous cells were treated in conjunction with other treatment options. This specifically refers to EB1089, which is essentially another analog of vitamin D3. When treated with this analog, there were the greatest results of anti-proliferative effects. Overall, this literature did consist of some strengths and some weaknesses. Overall, the study included a wide variety of test interventions and different analogs of vitamin D3. In total, there were 6 forms of vitamin D3 discussed in this systematic review. Now, this will add to the reliability that vitamin D3 has as a whole. Now, despite this, there were no human trials conducted up to this point. This is the primary weakness of the systematic review. And going along with that, there was only one study which involved live specimens, which were the mice that we had talked about earlier. Overall, this study can guide researchers to finding a new and promising treatment options for those who suffer from breast cancer aside from traditional chemotherapy and radiation treatments. Ultimately, within this systematic review, I found that vitamin D3 and its analogs have shown promising effects on breast cancer cell lines. There was evidence of cell death and the reduction in cell growth and division. Before I conclude this presentation, I would like to give a special thank you to Dr. Gerald Schaefer, as well as the Carroll College Health Sciences Department for their continual guidance and support in my research and education. Thank you all for taking the time to attend my presentation.